Hello students, welcome to EPG Pathshala. I am Mohan Chandra Joshi, an assistant professor from Jamia Millia Islamia. Today we are going to talk on modules, binary conjugates, selection schemes under the paper Genetic Engineering and Recombinant DNA Technology Part 1. After completing this module, you would be able to understand Introduction to Ti Plasmid mediated transfer of tDNA, through which agrobacterium infects the plant cells and causes tumor formation. You will also learn how conjugate and binary vector systems are made and being utilized by researchers. And in the end, you will get an overview how vector-mediated modifications to the plant genomes are done and how transgenic plants are made. Here is an example how Ti Plasmid introduces or transfers its tDNA into plant genome. In this picture, on the left hand side, you see a agrobacterium in which you have a genomic DNA copy. You have a plasmid, Ti type plasmid in it. Now, if a plant gets wounded, it releases a phenolic compound. This bacterium resides in the soil, it's a soil bacterium. So it can sense that wounded site by presence of a phenolic compound. Now, this bacterium can go and accumulate at that wounded site, infect the plant, and then can introduce that tDNA into the plant genome. As you can see in this picture, once the tDNA is inserted into the plant cell, it can induce tumor formation. And this happens because tDNA contains genes such as auxins and cytokinins, which can result in uncontrolled growth of plant tissues and result in a tumor formation.
These Ti plasmids cannot be used for genetic manipulation in vitro system to generate transgenic plants, and that's primarily because their size is too big. A natural occurring Ti plasmid is almost 200 kb in long, and also it contains genes such as auxin and cytokinins, which can induce tumor formation. Also, it also contains opine, which is generated in response to the infection of agrobacterium, and that can reduce the yield of transgenic plant generation. And lastly, the Ti plasmid contains tDNA, which is naturally formed or naturally occurring, and it cannot be manipulated. So in an in vitro system where we want to generate a transgenic plants containing our gene of interest is not present in a tDNA.
Modified version of Ti plasmids. Researchers have exploited the T1 type plasmid characteristics, which allows it to transfer tDNA into plant cells for their own use. So the researchers have found out that there are two regions of the Ti plasmid which is required for the efficient transfer of tDNA into plant cells. One of that is tDNA sequence itself, and the other is virulence region, which is required for the transfer, which encodes for proteins that helps us step-by-step transfer of that DNA into the plant cell. Using these two characteristics, researchers have developed two vector systems. One is conjugate based system, in which is a first generation one and not commonly used nowadays, but it is a single plasmid based system which is developed by recombining two plasmid having two different properties, and eventual products or the single plasmid that is generated in the end will be capable of transferring the tDNA, the modified tDNA sequence, into the plant genome. And the second system is a binary system. As the name itself means, it has two different plasmids, and these two plasmids are independent of each other, and both of these plasmids contain specific property that is required for the transfer of tDNA into the plant cell. And this system is widely used nowadays, and it's very common to see researchers using these systems to modify and generate transgenic plants in the lab.
Conjugate plasmid system is a single vector system which is used to transform into plant cells and generate a transgenic plant. A conjugate vector systems eventually contains all the information that is required for transferring tDNA sequence into a plant genome. However, this vector is generated by recombination method, and for that it requires two plasmid, namely intermediate plasmid and disarmed Ti plasmid. Now what is intermediate vector? Intermediate vector contains following information. It contains the repeat elements of a tDNA which is present at the border of the tDNAs. It also contains virulence region, which is a virulence apron. It contains origin of replication, and it contains a sequence which is from plasmid PBR322. This is a web plasmid of bacterial origin, namely E. coli. A disarmed Ti plasmid, which is also known as a truncated Ti plasmid, contains following DNA region. 1. Our gene of interest. Whatever gene we want to transform into a plant cell will be cloned in this plasmid. Will also contain antibiotics or a selection marker, and also it will contain similar, exactly same PBR322 sequence. Now these two plasmids will be transformed into agrobacterium simultaneously. After transformation, these plasmid, because they contain similar PBR322 sequence, will recombine and will give us a conjugate plasmid. Here is a schematic representation of how a conjugate plasmid system is generated. As I told you earlier, there are two plasmids that are involved in formation of a conjugate plasmid system. So very first one is a disarmed Ti plasmid, and in this schematics, as you can see, this is a disarmed Ti plasmid. It contains two tDNA repeat, left repeat and the right repeat, and which borders the E. coli PBR322 sequence. Now this sequence is going to be same in the intermediate plasmid also, and this disarmed Ti plasmid contains the virulence operon which is required for transferring this tDNA into plant cell, and virulence operon contains range of genes which are involved in various steps that are required for the transfer of tDNA. This is called disarmed Ti plasmid because it contains both tDNA as well as virulence operon onto the same plasmid, but rest of the information is being deleted compared to a naturally occurring Ti plasmid. That's why it is called disarmed Ti plasmid. It does not contain all the information of the natural occurring Ti plasmid, only required virulence gene as well as tDNA repeats. The second plasmid that is used for developing a conjugate plasmid vector is an intermediate plasmid. This plasmid contains an origin of replication which allows it to propagate it inside E. coli. It also contains a selection marker which is used in a bacterial system for its propagation. It also contains a plant selection marker. Now this selection marker will be used to screen the transformed plant cells once these plasmid is transformed into a plant cell. It also contains multiple cloning sites, and as I told you earlier, it also contains exactly same PBR322 sequence which allows the recombination between intermediate plasmid and the disarmed Ti plasmid. So this intermediate plasmid is relatively big in size compared to disarmed plasmid, and upon recombination this will make a single plasmid by replacing the PBR322 sequence here with the disarmed Ti plasmid sequence. So generation of conjugate plasmid system. So in the step 1 and step 2 we have shown you what is a disarmed Ti plasmid, which is a disabled form of Ti plasmid containing tDNA repeats and the virulence region, and also the intermediate plasmid which is containing our gene of interest as well as region of bacterial DNA PBR322 sequences which are used for recombination. And as you can see in this schematic, once these two are transformed into agrobacterium, because there is a homology region where PBR322 sequence can recombine and then they can form a single plasmid. So what happens in this case, the subsequent result is a conjugate plasmid which has all the characteristics including the modified tDNA region with all the repeats
as well as the virulence gene. The conjugate system, the conjugate vector system that eventually been used to transform is looks like something like this. It has a virulence region, as you can see shown in the red here, but it also have a scar region of PBR322 sequence. Because the recombination is not perfect, you still have some region of the bacterial DNA that is left. Then you have range of selection marker that were used, that will be used for the in the plant as a plant selection marker, which are usually in antibiotics. So once these DNA is transferred into plant cells, in order to screen whether the transformant having your gene of interest or not, these antibiotic selection marker will be used. Also, as you can see, there are two origin of replication present in it, and this eventual product is a large DNA sequence which will be used for transformation into the plant cell.
So what is the advantages and disadvantages of conjugate vector system? There are several advantages and quite a few are mentioned in this slide. One example or one advantage of this particular vector system is that the gene of interest can be cloned via recombination. Also, these plasmid is smaller in size compared to the naturally occurring Ti plasmid. So the smaller plasmid can be easily transformed into a plant cell to generate a transgenic plant. Also, the same plasmid can be propagated into a E. coli or any bacterial system to increase its copy number, and then can be transferred into agrobacterium, from where it can be used for infection. So this is advantageous in a sense that we have a capability to clone our gene of interest in a scar-free manner. Also, the plasmid size is smaller, and the same plasmid can be propagated into two different strains. So what are the disadvantages of this system? The major disadvantage with this system is that the sequence that is required for recombination is really big. So it is very cumbersome to clone this sequence or to have this sequence in a vector to begin with. And also the eventual plasmid, the conjugate vector system plasmid, is quite big in size. So what happens in that case, the efficiency of transformation into a plant cells is reduced because of its size. Binary vector system. This is the latest vector system that is available for transgenic plant generation. It's a dual plasmid system. Instead of having one plasmid like in a conjugate vector system, we have two vectors here, A binary and B helper plasmid. So the binary plasmid is a plasmid which is capable of propagating into both, again in E. coli as well as in agrobacterium. And binary plasmid contains our gene of interest. We can have with the promoter that we want in it, and the entire tDNA sequence is now replaced with our gene of interest. We can have both left and right repeats of tDNA flanking our gene of interest, though the left repeat is not important, or transformation can be carried out just with one repeat, that is right repeats of tDNA. We also need, in a binary plasmid, we also have a selection marker, which is again an antibiotics. And also this plasmid contains two origin of replication, one which allows its replication in E. coli, and second which allows its propagation or replication in agrobacterium. The second plasmid is helper plasmid, and that plasmid is already inside agrobacterium, and it can only propagate inside agrobacterium. It can't propagate inside E. coli. That plasmid contains virulence genes, which is required for the transfer of tDNA into plant cells. It also contains origin of replication, which allows it to propagate or replicate inside agrobacterium. Third, it does not contain tDNA sequence. It only contains the virulence factor, which is required for the transfer of tDNA, and it utilizes the information of binary plasmid to transfer it to plant cells. This is a schematic representation of a binary plasmid. As I told you in previous slide, it contains two origin of replications, one which allows it to propagate inside the E. coli, one that allows it to propagate inside agrobacterium. It contains two repeats of tDNA, right tDNA repeats and left tDNA repeats. In between that, we contain our gene of interest, whatever gene we want to introduce into a plant cell. It also contains the promoter, plant selection marker, which allows us to select or screen only those plants which are having our gene of interest, and also contain a bacterial selection marker. Again, that's an antibiotics which allows it to propagate into a E. coli or a bacterial system. Helper plasmid. Helper plasmids, as I told earlier also, these plasmids only resides into agrobacterium. Therefore, they contain only one origin of replication, which allows it to propagate it inside the agrobacterium. It contains virulence operon. Now, this operon contains range of genes which are required to transfer tDNA from the agrobacterium into the plant cells, and also it does not have any tDNA sequence in it. As I told you earlier, that tDNA sequence with our gene of interest is already present into the binary plasmid.
So what's the advantages and disadvantages of binary vector system? Let's look at the advantages. So the very good advantage and the very first advantage, as we can see in the binary vector system, is that we don't require a homologous recombination to happen. And as you remember from our previous slides, that homologous recombination required a large DNA sequence to be cloned in our vectors, which was cumbersome to begin with. Second is that vector size is relatively small. So once the vector size is small, it makes it much more efficient in terms of its transformation efficiency. And also the higher transfer efficiency makes it very easy to use. And also, being a smaller plasmid, it also helps you to manipulate it very easily compared to other larger plasmid, and that's what makes it a very favorite choice of researchers to make transgenic plant in the transgenic plant development.
So what are the major disadvantages of binary vector system? Binary vector system, the major drawback with this particular system is that the efficiency of replication initiation is different in both the host. As I told you earlier, it has two origin of replication. One that allows it to propagate inside E. coli, and second that allows it to propagate inside agrobacterium. However, the efficiency and the copy number of this plasmid varies in both the bacterial strain. Also, now having two origin of replication on the same plasmid also makes it a very unstable plasmid, and bacteria tend to lose such an unstable plasmid. Therefore, this particular system have this major drawback of having two ori in a vector. Though this is advantageous in terms of researcher because it allows the same plasmid to be used for two different systems and would not require subcloning of our gene of interest into a second plasmid, but same time having two origin of replication puts the system into a jeopardy. So how these vectors are used to develop a transgenic plants? In this schematic, we show how these vectors can be used to develop a transgenic plants. On extreme left, you see two vector system, conjugate vector system and binary vector system, in which we have already cloned our gene of interest. These vectors contain selection marker also, which are antibiotics, and now these plasmids independently, depending upon what vector system you want to use to develop a transgenic plants, you take that plasmid, transform into a plant cells using various transformation techniques. Once this DNA is inside the plant cells, it gets integrated into the plant genome. Now, we select these transformant by growing them in a selection media, and this selection media contains a selection marker which is already present on those vectors. Now, in presence of those selection marker, only transformants will grow and will give us a transgenic plants. However, these transgenic plants need to be further confirmed by several techniques that includes site-specific PCR, southern blot, as well as northern blot.
So students, let's now summarize what we have learnt in this module. Ti plasmids allow integration of tDNA into plant cells. This tDNA is modified by researchers depending upon the usage and depending upon their requirement. But tDNA transfer into plant cells, this characteristic was exploited from Ti plasmid, where researchers has concluded or found that there are two regions in the Ti plasmid, that is tDNA, and virulence region that is required for the efficient transfer of tDNA sequence into plant genome. Thereafter, they introduce a capability of modifying tDNA by various tricks, and then develop two different vector systems which are used nowadays to modify plant genome in vitro. So these two systems are conjugate system and binary system. Conjugate vector system is a one plasmid system. It has all the properties that is required for the efficient transfer into plant cells. It is results by the recombination of two different plasmid, the SAM plasmid and an intermediate plasmid, and which results in a large conjugate plasmid. The binary vector system consists of two plasmids. One is binary and another one is a helper plasmid. Binary plasmid can replicate inside both host, E. coli and agrobacterium, while helper bacteria can only survive or can grow inside agrobacterium. Once this is transformed, these two plasmid in combination can efficiently transform a modified tDNA sequence into plant cells. Therefore, by using binary system as well as the conjugate system, researchers can modify and generate transgenic plants. Thank you.